I'm very happy to give a series of lectures on my own preferred topic of research and teaching, namely bioinorganic chemistry. This bioinorganic chemistry is a very recent field of chemistry, and this field of chemistry was very young some 50 years back. Now it is a matured field. There are several books on bioinorganic chemistry, several journals coming out on bioinorganic chemistry. We have also worked on several aspects of bioinorganic chemistry in Bharathidasan University. And this bioinorganic chemistry is a very interesting subject because in this one we try to imitate the metallobiomolecules in biology. These metallobiomolecules, which you can sometimes simply say proteins and enzymes. These metallobiomolecules have very interesting function, and the function can be illustrated with the help of the structure of the metallobiomolecules. In fact, that is the very essence of chemistry itself, explaining the reactivity with the help of structures. So that's what we have been doing in our laboratory. We have designed a number of small molecules which would look like the metallobiomolecules, active sites, and we try to reproduce the function of these metallobiomolecules, that is, the reactivity of the biomolecules in a test tube, and try to explain them with the help of the structures of the molecules, small molecules that we make in our laboratory. So it is essentially, the subject itself is essentially an inorganic chemistry of the biological molecule. That is how inorganic chemistry is helpful in illustrating the function of metallobiomolecules with the help of the structures. Right. So I shall be giving a series of lectures. Today I shall take up one simple aspect that is in my lecture 1, that is biomolecular devices for electron transfer. That means I shall talk about metallobiomolecules which are involved in electron transfer. So we have heard about electron transfer in simple metal compounds or organic compounds. Here are biomolecular devices, that is proteins or enzymes, which are involved in electron transfer. So we would be eager to know what do you mean by this electron transfer function in these biomolecular devices. So we shall talk about it. Before taking up the subject, I would like to tell you that in the recent issue, very recent issue, which I received yesterday, it's Chemistry World, published by Royal Society of Chemistry. That is July issue, July 2020 issue. You can notice that there is a structure. This structure is an inorganic molecule which is trying to imitate the function of the protein molecule which is in the background. This protein molecule or the enzyme in the background is nothing but the nitrogenase enzyme. I think all of you know what is meant by nitrogenase enzyme. That enzyme tries to reduce the triple bond in nitrogen molecule, dinitrogen molecule, into ammonia, right? So that is the activity of nitrogenase enzyme. And here is an inorganic compound which people have made to imitate the function of the nitrogenase. You have heard about Haber's catalyst as iron oxide, which catalyzes the combination of reaction between nitrogen and hydrogen to give ammonia, right? So this particular compound is a synthetic analog, or we say synthetic mimic of nitrogenase enzyme. So that tries to reduce the nitrogen molecule. You can notice that the nitrogen molecule is coordinated to this complex. I shall be talking about it in detail in the next lecture, but then you can notice that this is a synthetic compound which people have become interested, right? So synthetic chemists, bioinorganic chemists are inspired by the structure of the enzymes which is responsible for the catalytic activity. We call that structure as the active site of the enzyme. So that active site is responsible for carrying out the enzyme reaction, right? So metalloenzymes have a big mastery. It's very difficult to reproduce the function of these metallobiomolecules, but we try to imitate the natural catalyst, and we try to learn a lot of things. So we take lessons from biology, and that drives us toward a new chemistry. So when we talk about bioinorganic chemistry, I have seen that this subject has given a facelift to inorganic chemistry. This has given a facelift to chemistry. Now people talk about bioorganic chemistry, like bioinorganic chemistry. People talk about biophysical chemistry. We put together everything nowadays, and people call it as chemical biology. So this chemical biology is a very new subject. That particular field of research has been started in several universities. Courses are being offered on chemical biology, and what I'm going to give, the series of lectures, is also a part of chemical biology. Particularly, I talk about bioinorganic chemistry. I shall take up, give you first of all an introduction about this metallobiomolecule. Here is a cartoon which represents the chemistry behind life process. What you see here is plants try to make carbohydrates, making use of photosynthesis. See, one can the plants use radiant energy from the sun and catalyze the reduction of carbon dioxide, the hydrogen atoms of water molecule. So carbon dioxide is reduced with components of water to give carbohydrates. These carbohydrates are generated, synthesized in the plant. When these plants are eaten up by animals and ultimately human beings, and the carbohydrates are broken down into again carbon dioxide and water. So again you have the same reaction to be performed again by biological system. So this photosynthesis and then the breakdown of carbohydrates, making use of the oxygen, which is a byproduct of photosynthesis itself, is very interesting. All these reactions can be now simplified as oxidation reduction reaction. So the very chemistry of life can be considered to consist of two chemical processes. One is reduction, the other one is oxidation. What is reduction? Carbon dioxide, right, that is reduced by the hydrogen atoms of water in the presence of catalyst and in the presence of light itself. Okay, so you have photosynthesis and you synthesize reduced organic compound, that is sugar molecule. These sugar molecules, in addition to this, you also generate a dioxygen molecule as a byproduct. The same dioxygen molecule is used in respiration to oxidize this particular organic compounds, carbohydrates, and you get back carbon dioxide and water, and you also get energy. So this reaction, the reverse of photosynthesis, is given here. Reduced organic compounds are oxidized with the help of molecular oxygen to get back carbon dioxide, and we reap energy, right? This reaction is not also very simple. It is also a catalyzed reaction. For example, we are made up of organic compound, right? And we are exposed to constantly dioxygen molecule, but we are not oxidized, right? But there are several histories. In history, there are several examples where instantly people have been oxidized to carbon compounds, carbon dioxide. So in this reverse reaction, energy is released. I would like to point out that these reactions involve thousands of reactions. This is only a summary, simplified reaction. Each and every reaction involves thousands of reactions. Thousands of bioinorganic compounds, metallo-biomolecule molecular devices, right? All of them are involved in concerted action. So people have not tried to still now discovered, unraveled the mystery behind photosynthesis at all. So these two reactions are simplified reactions. I shall tell you our existence in this earth depends on the ability of these plants to capture sunlight, right? If there is no capturing of the energy, sunlight, then we will be also extinct. So all these two processes I talked about, both oxidation and reduction, both involve metal ion. People say 70% of all the biological reactions involve metal ions. So people call, for example, biochemistry as bioinorganic chemistry, or simply sometimes people call that as the coordination chemistry of living systems. Most of the time, the metal ions will be associated with the metallobiomolecule. Sometimes metal ions are taken up whenever we want to reproduce the reaction, right? So the ability of you to employ catalyst for these two reactions, that is, you have to use controlled catalyst for the release of energy from the carbohydrate. When you get that energy in living systems is used to make ATP from ADP. I've given you the structure of ADP here, adenosine diphosphate. And this one combines with inorganic phosphate, right? So when it combines with inorganic phosphate, you get adenosine triphosphate, right? This adenosine triphosphate, you can notice that there is a bond that is formed here, PO bond. So the energy that we supply for making ATP is stored in the form of a phosphorus oxygen bond. See, people should realize now that how energy is stored in biomolecules. They are all stored in the form of a chemical bond. So this chemical bond is the way in which energy is stored. Now people can think about the storage of energy in industry. So they can store the energy in the form of this bond, right? In fact, people say 35 kilograms of ATP is synthesized in our own body every day, and that is being utilized. How it is utilized? This is particular, for example, this reaction is made possible in the mitochondria. Made possible in mitochondria, where this synthesis is done. That's energy generator. Mitochondria is called energy generator, and this synthesis, is also done in mitochondria, and hence we can say that this is an energy generator. And when this ATP molecule is taken up, is transported throughout our body for doing all sorts of chemical reactions. During that time, this ATP will be hydrolyzed. This bond will be hydrolyzed, and you get back the energy. So you get back the ADP and inorganic phosphate. So ATP is the energy containing molecule. People say it's the energy currency in biology. So ATP is the molecule which contains the energy, and so this reaction behind the ATP reaction or the dioxygen evolution in nature can be is performed by a metallobiomolecule known as the oxygen evolving complex. So I'm taking up this example to illustrate the role of or the scope of bioinorganic chemistry. We just look at the oxygen evolving complex, which is a manganese containing metallobiomolecule. There is an enzyme which is involved in photosynthesis, right? This enzyme, tries to decompose water. This decomposition of water is done is at a pH of 7, and we get dioxygen, and you are able to generate 4 electrons. So people should realize here that how electrons are generated in nature. So one can use this complex make this enzyme to generate electrons. So this generation of electrons itself is very interesting. For example, if you take this water molecule, try to take it in a test tube, put two platinum electrodes, try to decompose water by applying a very high voltage. You will be able to decompose water at high voltage. You get hydrogen and oxygen, but they are all mixed. So you cannot get pure hydrogen, pure oxygen. You need a lot of energy to break the OH bond in water. But what happens in nature is at a pH of 7, at ordinary temperature, right? At ordinary temperature, namely room temperature also, this water is decomposed by the oxygen evolving complex in plant. See, this is how electrons are generated. This is how the byproduct, namely dioxygen, is generated. Now look into the chemistry behind this one, or the inorganic chemistry behind this reaction. It is a cluster catalyzed reaction. The oxygen evolving complex, that is the enzyme, is said to consist of a manganese, a dimeric manganese. Some people say it's tetrameric manganese. We have a very simple theory like this, where two manganese ions are put together like this, bound through oxo bonds like this, and this particular structure also has two water molecules bound to this. This is what we call as the active site structure of the oxygen evolving complex. So this structure, active site structure, is responsible for the decomposition of water. And it has been proposed that when water molecules bind to manganese like this, the normal oxidation state of manganese is 2+, but this contains higher oxidation state of manganese so that it can easily bind to water. You notice that the two water molecules come together and form a peroxo bond, and you generate 4 electrons. This reaction is a very fundamental reaction. How 4 electrons are generated. The reverse reaction is also important in biology because the dioxygen can be reduced by electrons to give water. Sometimes the dioxygen is reduced to get hydrogen property. So this is a very fundamental reaction in biology where electrons are generated. So now you see these 4 electrons are generated making use of a metallo-biomolecule. So this we call it as a cluster catalyzed reaction. Okay? So this peroxo bond will be decomposed. You have generation of water and then dioxygen. So this is how now students of chemistry should understand how oxygen becomes the byproduct of this photosynthetic reaction. So water splitting, that is the most important thing. So a lot of people throughout the world now are making compounds containing manganese, compounds containing cobalt, compounds containing copper to decompose water at low energy. By giving low energy we can try to decompose this OH bond in water. So people are trying to reproduce this reaction. Still now, no structure has been made, no small molecule, bioinorganic molecule has been made which can perform this decomposition of water molecules to generate 4 electrons. So once you have a low molecular weight bioinorganic model compound, a catalyst. So you can generate a catalyst now. If you have a catalyst like this, you need not bother about the lithium energy battery. You can generate 4 electrons with this manganese containing compound by putting that in water. See, this is how people say, no, you can run a car, you can run a bike with the help of water. So now understand, this is the secret behind this one. When this hydrogen plus combines with electron, you generate hydrogen. So this hydrogen can be stored and you can use that as a fuel model. So nature, now I have shown you how you learn lot of lessons, right, from probing the natural reaction, right? So you can make a manganese catalyst and try to generate electrons or generate hydrogen molecule. People have made also nanometallic compounds, whereby using which you can decompose water at low energy, right? So now we can remember that there are a lot of things you learn from nature, and you can take chemistry in newer horizon. So that is called chemical biology. You use your chemical knowledge, chemistry knowledge, to explain these biochemical reactions, and you try to learn lessons. You try to make this basic science and convert that into a technology, right? So this is how people try to do a lot of other things also. Researchers try to transform biomass into energy-rich fuel molecules. Once you have photosynthesis and generate these green plants, these green plants themselves have carbohydrates stored. So you can grow these small plants, try to extract the carbohydrates in the plant, and then make use of them as fuel molecule. So Giant Bioenergy Institute in the University of California, they're trying to grow plants and use plant cells and extract the carbohydrates from the plant cells. But plant cells are recalcitrant. They don't give their carbohydrate easily. It's very difficult to get back carbohydrates, sugar molecules from these cell walls, right? They are all very complex polysaccharides, right? Here is another thing which we learned from this oxygen evolving complex or photosynthesis: carbon capture and usage. I told you just now how carbon dioxide can be reduced with water molecule to generate carbohydrates in nature. Now people are wondering, why do not you reduce carbon dioxide suitably with hydrogen to make methanol? So once you convert this carbon dioxide, a greenhouse molecule, which you give out by burning your petrol, diesel, etc., they can be, converted into carbon dioxide in these molecules from burning carbohydrates. They can be converted into methanol itself. For example, if you take methanol, get methanol from natural gas, it costs $400 per metric ton. But when you try to synthesize methanol from carbon dioxide by reducing it using a catalyst, then it costs higher, $1,500 per metric ton. And you can also try to make formic acid from carbon dioxide. So we have learned lessons from biology, how carbon dioxide is reduced. In fact, people have tried to make a solar mat, that is a synthetic leaf, where you can try to pass hydrogen over carbon dioxide, reduce it to methanol, and then pump it out. So this is the catalyst people are trying to make as a synthetic leaf. A polymer mat, which is made of a silicone fluoropolymer, is constructed like this. You have a grass-like array of micro wires made from the silicon, and this nanometer thick layer of this one has a nanometer thick layer of titanium dioxide coated on this one, and that is the nanomaterial which contains nickel substituted into this titanium dioxide that acts as the catalyst, right? So when you pass hydrogen to carbon dioxide, see, you can, instead of this you pass carbon dioxide and water molecules, then carbon dioxide will be reduced, right, to give useful chemicals. So this Carbon Recycling International has developed an efficient low temperature catalyst which turns hydrogen and carbon dioxide into water and methanol. Methanol can be distilled and water can be recycled. Okay, so this is a very good example to say how a synthetic leaf can be made by reducing carbon dioxide. So this is how people are progressing, and we have learned a lesson. We have learned that nickel can be used as a catalyst, and nanomaterial can be used as a catalyst to make this type of catalyst to reduce carbon dioxide. Now let me take up this metallobiomolecule, that is how metallo-biomolecules, which are also called proteins, how these metallo-biomolecules contain the metals coordinated to the biomolecule, namely protein. So proteins are the metallo-biomolecules which take part in this catalytic reaction. So we should first of all know how these proteins bind to the metal. And before that, we should also notice that the metallo-biomolecules have very interesting functions. The functions are very efficient, very complex, and it is difficult to understand the mechanism also. And this particular, reactions, functions which I am going to talk about, there is no parallel in the chemistry field, the field of chemistry. For example, we take redox and catalytic reactions. And this biomolecule, which I mentioned here, they have no parallel in a test tube. You cannot duplicate these reactions at pH of 7 in aqueous condition. For example, here itself you can see you have an enzyme which represented here. You have an enzyme which contains a catalytic component and a redox component. That is, this component will supply electrons for carrying out reduction reactions by catalyst. So this is a very simple way by which you define an enzyme. This contains a redox component. So this involves electron transfer. So this metallo-biomolecule, even though they do catalytic reaction, they should supply electron. So these reactions catalyzed by the enzymes, they have no parallel in biology, sorry, in chemistry. For example, you can see here, this is the hemoglobin, right, molecule, very complex molecule. It's a tetramer. So each and every component, we call it as alpha 2, beta 2, has a porphyrin, iron porphyrin coordination compound. Four are there. So they are all, well preserved, well protected from the aqueous environment. So these catalysts here, this particular porphyrin ring structure can take up molecular oxygen. So they are all protected from the atmosphere, these catalysts, right? So what is the function of this organic molecule, organic component protecting this inorganic compound? So these inorganic compounds, they are also protected from the organic compound. So we say the protein environment, here also another enzyme reaction, right, where a porphyrin ring system is protected by the organic molecule, enzyme molecule, peptide molecule. So these protein molecules have a very complex environment. They try to save and stabilize the coordination compound here. So this coordination compounds, that is metallo-biomolecules, are nothing but enzymes and proteins where you have an inorganic component, coordination component, well preserved by the protein environment. It is the protein environment, organic environment, which imposes the ion to have a very novel geometry, coordination geometry. These geometries are very unstable in a test tube, in aqueous solution. That is why I say there is no parallel for this reaction. Either you take molecular oxygen uptake by this hemoglobin or electron transfer or catalysis, they have no parallel in simple chemistry. That is why I said this learning of metallo-biomolecules will take us to a new horizon in chemistry, right? So we understand a lot of principles. So what is the role of a bioinorganic chemist? As I said, the gas of oxygen evolving complex, you investigate, probe the nature. Like people say, man looked at the bird, how it flies, and made aircraft, right? The same way, you try to probe everything, atoms and molecules, and try to search and study things created by Mother Nature, and use the knowledge that you get for human application. That is now what I told you, how electrons can be generated. Please do not depend on lithium. The amount of lithium in the world is decreasing. So one day you may not have lithium. So now you have a solution how you can generate electrons by learning from nature. So we can now, people talk about corona and all, you can now learn a lot of things from biology and try to deal with them, right? For human applications, you can now generate, for example, I told you about manganese compound, you can generate a catalyst by bioinorganic study and use that as a catalyst for performing the novel reactions carried out by metallo-biomolecular. Right? So before trying to take up one particular metallo-biomolecule and then wonder at how electrons are transferred. That's today's lecture. One should know how to study these metallo-biomolecules. These metallo-biomolecules can be conveniently classified into proteins and non-proteins. Well, proteins contain an amide bond, right? Non-proteins, they don't contain an amide bond. So these proteins can be now classified depending on the function of the protein. When these proteins, are involved in transport and storage of electrons, storage of electrons, transport of electrons, transport of proteins, transport of metals, transport of dioxygen, right? So all these will be, called transport. This transport is done by the metallo-biomolecule, namely metalloproteins. When these metalloproteins take part in catalytic reactions, we call them as enzymes. We shall talk about these enzymes in a later lecture. Now let us take up these proteins which perform, the transport function, storage function. This can be further classified depending on the metal ion present, depending on the type of function. For example, you have metalloproteins which take part in transport of electrons, electron carriers. When they are taking part in electron transport, we call them, we can just further classify them depending on the metal employed by the protein. If nature makes use of copper, then the metallo-biomolecule is called blue copper protein. In fact, today I shall talk about this blue copper protein and illustrate how electron is transferred by this blue copper protein. This blue copper protein is present in green plants. In addition to this, we also have iron sulfur proteins. They contain iron. So not only copper, even iron is also employed. The beauty here is that the copper and iron can exist in more than one oxidation state. Copper 2+, copper plus, iron 2+, iron 3+. Just because they are able to exist in more than one oxidation state, they are used for settling electrons, for taking up electrons, for storing electrons. So nature has very conveniently used only copper and iron metals for making up metallo-biomolecules for electron transport, right? So these proteins, when they store iron, for example, in large quantity, we will take it up in a later lecture. People say, for example, ferritin is a biomolecule, it's a protein which stores 4,500 iron atoms in a particular molecule. It's used as a metal storage protein. Ceruloplasmin is a protein which stores copper. So copper and iron can be stored. These are needed to synthesize our proteins and enzymes. So they are synthesized then and there. They are available in cells and human plasma. So they are utilized to make these biomolecules, metallic biomolecules, and they are utilized for the storage molecule. And sometimes the metal ions can also be used to maintain the novel geometry of the metallo-biomolecule. So This transport is another important function of this metallo-biomolecule. Another important function of the metallo-biomolecules as a protein is the signal transduction. We are not going to consider this today. I shall now consider another type of transport protein. You have a family of proteins which are involved in dioxygen transport, dioxygen binding, right? So the dioxygen binding, oxygen, I told you, it is produced in nature itself as a byproduct in photosynthesis. So that oxygen molecule is taken up by human beings. And when it is taken up in respiration, in lungs, they are taken up all over our body. They are stored in myoglobin. Hemoglobin functions as the transport protein, and we also have another type of metalloprotein, iron-containing protein, which use a different principle for dioxygen binding. So notice that depending on the metal, the principle will be different. For electron transport, storage, and oxygen binding, different principles are used. The dioxygen binding itself, you have three different, principles used. Hemoglobin uses one principle. Hemerythrin is another molecule present in some other species. They are using a dimeric ion. So these are the three families used in nature for binding dioxygen, and then using it in mitochondria for binding our. So it is very interesting to see how these metallo-biomolecules can be classified and what type of metal ions are utilized for all these functions. One can also start from copper and then wonder at their functions like electron transport, metal transport, dioxygen binding, and then end up with a metallo-biomolecule. So I have conveniently used this way of classifying the metallo-biomolecule, study them in terms of principles involved in the metallo-biomolecule function. So today I shall take up, as I said, the electron transport by these proteins. Today I shall enumerate what are these blue proteins and how electrons are transferred. Before that, you should also know how these proteins are able to bind metal ions. So proteins are used as ligands. Metals, whenever people say metal is there in biological system, immediately one should imagine that it is present as a constituent of proteins. Okay? People are isolating newer proteins nowadays. And when you say you have metal protein, immediately you should figure out that that metal ion is coordinated to the organic molecule. So it's a good question how metal ions are bound to proteins. You know very well that a protein is made up of these amino acids, this repeating unit, right? All these amino acids contain the R group, side chain group. Okay? When this R group is different, you have different amino acids. When the R group is CH2S methyl, the enzyme is called methionine. The interesting thing is this methionine has a sulfur. You know very well that sulfur has two lone pairs. This lone pair can be donated to a metal like copper or iron. So any protein which has a methionine will offer its sulfur for coordination. See, sulfur is a good ligating atom. We now talk about the soft donor or soft metal, hard metal like that. Okay. When the R group is CH2SH, then we call it as cysteine. Cysteine contains, a mercaptan SH. So H can be its acidic that can be replaced by a metal like copper and iron. You have, cysteine as a very good ligand. It offers H bond for coordinating to metal. Similarly, you have histidine amino acid. When the R group is CH2 imidazole, right? This imidazole contains two types of donor atom. We say it is a polydendrite ligand. How? So it contains nitrogen, which is very similar to your heterocyclic nitrogen in pyridine. So you have a lone pair that can be donated to metal like copper or iron. So it is also a very good ligand donor atom. In addition to that, this has an NH group. We say it's a pyrrole NH group, right? So this can also be made acidic. This at high pH, you can remove this and you generate a lone pair which can be donated to metal. So you see that this ligand component, a ligand moiety of histidine amino acid, can function as a bidendrite ligand, a mu ligand, right? So whenever the pH is high, you can try to use this as a ligand. You must be knowing now that this imidazole, simple imidazole itself is used as a buffer by biology. It can be acting as an acid or as a base. So nature uses this to coordinate to metal, and this can also coordinate to another metal. For example, superoxide dismutase, copper, zinc, superoxide dismutase. Copper is here. This can be deprotonated, and you can have zinc coordinated. So these are all very interesting ligand system. In fact, histidine is the most interesting, most omnipresent, most omnipresent donor atom to anchor metal ions to proteins. You take any metalloprotein, you will see that this histidine is present as a moiety for coordinating to metal. So we also have another interesting amino acid, tyrosine amino acid. So when the R group is aromatic compound like this OH, phenolic compound, this metal, this OH group can be deprotonated and you can introduce iron or copper. I'm going to tell you about a lot of metal biomolecules where iron and copper are bound to this OH group. So tyrosine is an interesting amino acid. See, you can see phenol is present there, but phenol is not different. Phenol can be poisonous, but this tyrosine is not poisonous. This will be deprotonated only after a high. So all these proteins with side chains like this, they act as ligands to coordinate to metal ions. Nature has, in addition to these ligands, a very interesting, molecule, that is porphyrin ring system. Porphyrin ring system itself is demands separate study. People have published books, several volumes of books on this porphyrin ring system, right? So this porphyrin ring system is one particular macrocyclic ligand that can be used as a polydendrite ligand. Please note that this nitrogen has a lone pair, this nitrogen has a lone pair, this can be donated to metal. It also has two pyrrole groups. You can try to remove hydrogens and make it acidic and introduce metal ions like copper or zinc or nickel. So this can be functioning as a tetradendrite ligand. This ligand is very interesting because nature makes use of different forms, different substituted forms of this macrocyclic ligand. When you introduce an electron donating ligand here, the donating substituent here, this will be made more basic. So this will be a better donor atom for metal. When you use an electron withdrawing group, a vinyl group in hemoglobin, this will become acidic and you can replace hydrogen by a metal like iron. So this can function, this porphyrin ring system can function as an acid or as a base, and this will function as a tetradendrite ligand. You can introduce copper or iron, make very stable complex. So remember, nature makes use of a very interesting one ligand, one particular ligand type, but differently substituted ones can be generated, and you can generate different ligand system. So nature doesn't make newer ligand system to make newer metal biomolecule. No, they make use of one particular ligand like porphyrin and where is its donor capacity, where is their coordinating capacity, and introduce any metal ion you want. Depending on the reactivity, depending on the type of reaction, function of the metal biomolecule, you can introduce different substituents. I shall take up in a later lecture how this functions as a very interesting ligand. In addition to that, I should also mention at this point, this is a very delocalized system which can take up electrons and store that. Not only it is a ligand, it is a delocalized, stable organic system which can take up electrons. You can reduce ion coordination compound of this. That is a heme group. We can add electrons to this, right? Here is an example. Hemocyanin. I told you about hemocyanin, copper containing one, which is used for dioxygen transport. Here is the active site, which binds to dioxygen. So it is the organic matrix which imposes upon these two copper atoms to have a very interesting coordination geometry. This has two copper and two oxygen atoms bound like this. People have been wondering about how oxygen is bound to hemocyanin and transported in small species like in small snails and other things. How do they transport molecular oxygen? And a particular scientist like Kitajima in Japan, he made a compound, coordination compound, looking like this. And he tried to synthesize a compound in which he was able to trap molecular oxygen. He found that molecular oxygen is trapped between. This we call it as a very interesting bioinorganic compound where molecular oxygen is imprisoned between two copper. So this is a very important bioinorganic discovery. He illustrated that metalloproteins, metallobiomolecules bind to oxygen molecule and make use of oxygen molecule for several reactions also. So this is a very big breakthrough in bioinorganic chemistry, how dioxygen is imprisoned between two copper atoms. So we should remember that by trying to study this metallobiomolecule, by trying to imitate these biomolecules, not only that, you try to explain these reactions carried out by the biomolecules, the interesting structures, and relate them to their functions. Also, you try to imitate. That is the envy of any synthetic chemist. You try to make newer molecules. As in nature, try to use this in technology, right? So this is just a slide which illustrates how these proteins are, having a linear structure, how the amide bond is made, and actually this is a linear structure, it's made up of amino acids. This linear structure is in fact, having two structures like alpha barrel, beta barrel. So you can have a linear structure like this, right? This linear structure is stabilized by hydrogen bond, right? So these linear structures are called barrel structure, alpha and beta 1. This is this can be right-handed alpha barrel, it can be left-handed beta barrel. So these are fundamental organic molecules having hydrogen bond. Thousands of hydrogen bonds make up this one. And these linear structures, barrel structures, they fold again. They fold again, right? They fold and stabilize a tertiary structure here. These tertiary structures, are able to stabilize the bioinorganic active site, the heme structure here. This is the one involved in dioxygen uptake. So this is stabilized by protein folded structure, right? So interesting catalytic or reactive structures, inorganic structures are stabilized by the organic mind. And you can notice that this folding helps to stabilize our inorganic mind. In fact, I have heard lectures by our Professor P.K. Ponnusamy, who was our in Bharatidas University head of the Department of Physics, and he is a protein folding biophysicist involved in how proteins are folded. He used to tell me an analogy how these proteins are tempted to make folding. For example, when we go out in the cold, during the cold season, when we have cold wind, what we try to do, we put our hands together and try to save our lungs and heart. We fold our hands to protect our active site, namely lungs and heart, which are doing the function of molecular oxygen uptake, circulate blood circulation, everything. So that tendency is there in the proteins that we have, that is the hemoglobin we have. So I always remember this analogy. If people are able to visit his house in Ponnagar in Trichy, please try to notice that he has made all his house, the windows and everything, the grills, he has made it in the shape of this biomolecule. How oxygen is bound to this biomolecule. Interestingly, all these grill works he has are made up of the fundamentals of this protein folding. One can enjoy visiting his house and learn about dioxygen, how dioxygen is bound to these proteins. Right. So let us take up now today the electron transfer function. I told you there are a number of metallo-biomolecules containing copper and iron which are involved in electron transport. So we talk about wires, right? What do these wires contain? The copper and iron and aluminum. So nature uses copper itself to deal with these electrons, and we use also iron for electron transfer. So we have proteins, that is organic components, which impose certain types of structures on copper and iron, and ask that iron and copper to get involved in electron transfer. So electron transfer proteins participate in the various functions which are essential to foundations of life. What are these foundations of life? Respiration, photosynthesis, nitrogen fixation, biosynthesis. Any biosynthesis you take, biomolecule, synthesis of biomolecule, it involves electrons. Metabolic processes, dioxygen metabolism, how carbohydrates are burnt with molecular oxygen. Take, for example, nitrogen fixation, how nitrogen molecule is reduced with electrons from, again I should say, from the oxygen evolving complex. So they are only used for reducing nitrogen to the ammonia, which is used as manures. Ammonia is a basic molecule for making manures for plants. That nitrogen will form the components of amino acids, right? Same way respiration, how electron is carried in our body. We take molecular oxygen and how it is involved in burning of food. And there is a chain, which is called Britten-Chance chain of electron transport. So electron transfer proteins participate in very important reactions. So as chemists, we should know how this electron is transferred very easily in a very facile way. We have a question: how can these facile reactions can be reproduced in a test tube? So with that idea, we shall now look into the structure of this metallo-biomolecule and try to explain the function of these metallo-biomolecules with a structure. So if you want to study any particular reaction, you should first know the reaction. That is called structural biology, or that is use the structures of metallo-biomolecules to illustrate the function, or the modern term is chemical biology. So we have a lot of examples for electron transfer proteins, which I shall take up in a series of lectures. One is blue copper protein. how copper is able to transport electrons. And we have iron sulfur protein, right? This is a cluster containing protein which makes use of a different principle for electron transport. Blue copper proteins use one type of transport. We have mononuclear copper, whereas here have multinuclear iron cluster. We also have a third type of proteins, that is cytochromes. They also have iron and they have the porphyrin ring system. They are mononuclear. Iron can use it as a multi-ion system, or we can use a mononuclear system. See, this mononuclear system has an iron protein, iron coordinated porphyrin ring system. The same type of iron porphyrin system can be used to bind to molecular oxygen in hemoglobin. The same type of system can also be used as a system to carry electrons for doing all these reactions. So nature makes use of three different principles for transferring electrons in a very facile fashion. That also depends on the type of living organism. We make use of cytochromes in respiration, and iron sulfur proteins are used in plants. Blue copper proteins are used in proteins in plants. So these I can summarize the importance of electron transfer proteins. Okay, let me take up the blue copper proteins. It's a biomolecular device for electron transfer, right? So where does this occur? It occurs in plants, and the electron transport or photosynthesis synthetic scheme in plants can be summarized with the help of the so-called Z scheme here. This is the Z scheme. So this Z scheme is nothing but a plot of the redox potentials of the various biological systems, metallobiomolecules involved in electron transfer, right? So this is a simple theory, proposition behind the photosynthesis. There are two types of chlorophylls. One chlorophyll is in photosystem 2 that absorbs light 680 nanometer, right? Another one, P700, absorbs light at 700 nanometer. There are two photosystems present in photosynthesis. So what does it do? It just absorbs sunlight, right? Sunlight, and tries to, excite electrons. So this gets electron. Where does it get electron? The water molecules. It's a manganese enzyme, oxygen evolving complex, right? Which decomposes water at low energy to generate 4 electrons here. It is these 4 electrons which are energized by these photons. So they are now taken up by organic compounds, quinones, A, quinone B, passed on to plastoquinone, and then these cytochromes. There are large number of cytochromes, complex cytochromes present. Then pass it on to iron sulfur proteins, pass it on to cytochrome F, then to plastocyanine. So this plastocyanine involves one of the redox partners involved in electron transport in photosynthesis that carries the electron to the reactive center and reduces carbon dioxide. So this is not a simple reaction. Photosynthesis involves large number of redox partners. So what do? They energize the electron. You can have an analogy. Suppose you want to go from ground floor to first floor. How do you go? You don't go in one jump. You use stairs, several stairs, right? One by one. Like that, several proteins, redox partners are employed to energize the electron. Okay? These electrons, these small steps are used to energize the electrons in a thermodynamic way, reversible way. So these electrons are energized. Again, it is energized in photosystem 2, and these are used to make organic reductants, NADPH. This is reduced like so. This is a direct way by which the energy is used. There is another way by which the electrons produced are employed to make ATP from EAD. So now realize that the redox partners are very important in photosynthesis, right? So what are these blue copper proteins? What is the novelty behind the use of these blue copper proteins in electron transfer? As I said, the function of these proteins is electron transfer in photosynthesis. One of the redox partners taking part in electron transfer. So how is it able to function as electron transfer protein? That originates to the novel coordination geometry around copper. That is, it has a novel mercaptan bond, copper to mercaptan bond. This is cysteine bond, amino acid. So copper to mercaptan bond is stable. Normally in a test tube, if you add any mercaptan, mercaptacetic acid or lactic acid to copper to in a test tube, immediately the copper color will be removed, right? That's copper 2 will be reduced to copper 1. But in these proteins, the copper mercaptan bond is stable. Simply, that is because of the novel coordination geometry. When you add a mercaptan to copper, the bond that takes up the orbital that takes up the electron is the x square minus y square orbital, which takes up the electrons easily. But here, the x square minus y square orbital in this novel coordination compound is not oriented towards the x-axis or y-axis. So how do we know about all these things? By making models for this blue copper protein. So in our lab in Bharatidas University, we have made a lot of compounds and try to wonder at how these proteins are able to engage in electron transfer. We have tried to point out that it is the mercaptan bond which is stabilized in the protein. This cannot be stable in a simple test tube. For example, the MSc students, you make tris-thiourea copper. How? Thiourea can be considered as a mercaptan that enolizes. Yes, it form a SH bond. That is used as a reducing agent to reduce copper 2 to copper 1. So you make excess thiourea that binds to copper 2, that is copper 1 generated, and you get tris-thiourea copper. It's a good example to illustrate that copper 1 oxidation state is stabilized by sulfur containing ligand. But in proteins, these bonds are not, this bond is stable. That is why I say all these blue copper protein or any metallo-biomolecules are very interesting because they have an abnormal coordination geometry. So when you say they have an abnormal coordination geometry, they also have abnormal spectral properties. You can compare these spectral properties here. These spectral properties cannot be imposed in simple copper compounds or simple iron compounds. One important property is these blue copper proteins, so-called blue copper proteins, have an intense visible absorption band at 600 nanometer. What is the epsilon? 3000 per mole. So it just acts as like a dye molecule. Organic compounds are known to have very high epsilon, right? They are very excited easily and become colored. Same way, these blue copper proteins have an absorption at 600 nanometer and they have very high ease of excitation. What is epsilon? That is the ease of excitation. In simple copper compounds, also you can excite the electron. You can have a transition from T to G to EG. That involves 600 nanometer, but the ease of excitation is very low. Epsilon is 10 per mole only. That means the epsilon is very low. So this particular 600 nanometer, is the reason for the visible absorption, and hence the color of the copper protein, that is blue copper protein. We also call this blue copper protein as a type 1 protein. The later lecture I shall explain what is meant by type 1 copper protein. So this is the novel property of blue copper protein. This blue color cannot be generated in simple copper compounds with so much intensity. Even if you have microgram, milligram of this blue copper protein, they will have a blue color, right? So these copper complexes will have very thin color because the epsilon is only 10 or 8 or the maximum 100. That is how these blue copper proteins are different from our simple copper compound. Another important property is, the EPR characteristic. That is EPR spectroscopy. That has a parameter called A parallel. This A parallel for our blue copper protein varies between 30 to 70 Ga. Whereas a normal copper compound, copper sulfate compound dissolved in water, for example, or in methanol, will have an A parallel value of 140 Gas, 200 Gas. So that is how this A parallel for blue copper protein is different from the normal copper compound. Why? This A parallel is always a measure of the interaction between the unpaired electron on copper with the copper nucleus. When the interaction is strong, the A parallel will be very high. When the interaction is weaker, it is low. Now, this A parallel is also dependent on the coordination geometry of copper. When copper has an octahedral geometry or square planar geometry, this A parallel will be 200 Ga. Whereas if you have a tetrahedral geometry, the A parallel will be 70 Gas. Now you see, you have a way of distinguishing the coordination geometry around copper compound. This blue copper protein now has a copper with a tetrahedral geometry. So these properties, spectral properties, can tell us what type of geometry is there for the active side, which is responsible for the electron transfer, is present in blue copper protein or any protein. So A parallel is 30 to 70 Gas. That is the novel spectral property. One can use this A parallel value to distinguish blue copper protein from another copper protein. To distinguish this blue copper protein from normal copper compound. The most important property, the next important property is the high copper to copper redox potential. This is how we talk about the electron transfer with this parameter. See, this particular copper proteins have very high redox potential. For example, the E half for this copper to copper potential, copper potential blue copper protein is 0.4 to 0.7. You can notice in the Z scheme that the plastocyanin is a good example of blue copper protein, has a redox potential of 0.4 volt, right? So these blue copper proteins have high redox potential. So when you substitute that value, E value in the Nernst equation, you see delta G is negative. This means that the redox in blue copper protein is very spontaneous. The electron transfer is very facile. On the other hand, when you talk about the redox potential of normal copper compound, like aqueous copper, which has 4 water molecules or 6 water molecules bound to copper in a square planar arrangement or an octahedral arrangement, there the redox potential is more negative. When the E value is negative, now you substitute here, when E is negative, delta G will be positive. This means the electron transfer by copper complexes, simple copper, very small, tiny, but you need lot of energy, lot of potential to transfer electron to this copper in small molecule. The reduction is very difficult. That is why the redox potential of aqueous copper is more negative. You take any aqueous compound or any complex compound, that will have a very negative value. Whereas these blue copper proteins have a positive value. That is how these blue copper proteins has a novel property which is different from our novel copper compound. So we have abnormal properties. These abnormal properties is difficult to be reproduced in a test tube, right? So we can now just consider some blue copper proteins. We have fungal lacase, tree lacase, celluloplasmin, azurin, plastocyanin, stellocyanin, plastocyanin. All of them are the copper proteins. What about the redox potentials? What about the color? They are blue. What about the redox potential? Very positive. See, that means they can very easily transfer electrons. Now compare aqueous copper. With respect to normal hydrogen electrode, this aqueous copper has redox potential, copper 2, copper 1 potential 0.15 only with respect to normal. So it is having less positive value. That means electron transfer is facile in blue copper proteins rather than in aqueous copper. Now we understand why reduction is difficult in aqueous copper. And you can use Nernst equation simply to explain the fast electron transfer taken up by blue copper protein in green plant. So I told you about the blue copper protein. This is the electronic absorption spectrum of the blue copper protein. But normal compounds have a very low epsilon, whereas blue copper proteins have a very high epsilon, very strong absorption, which imparts blue color to the blue copper protein. Here is the blue copper protein EPR spectrum. This is the normal EPR, and we define this gap as the A parallel. See? We have four hyperfine lines, and the distance, you can take the distance between these four, divide by 3, you get the A parallel. So that A parallel will be 150 Gauss as 164 waves per centimeter, 10 power minus 4. Simply you can call it as approximately 160 Gauss. So this contains two components, parallel component and perpendicular component. Same way, blue copper proteins will have a low A parallel value. And a G perpendicular, you have a center that is G parallel value. So you can find out two parameters to blue copper protein EPR spectrum. One is the distance here, that is 63 Gauss or 70 Gauss, right? So you can measure this distance. So you can distinguish very well a blue copper active site structure from normal active site, copper site. This has a square planar geometry, whereas this has a geometry which is very close to tetrahedral. I told you tetrahedral copper compounds will have a low A parallel value. Square planar compounds will have a very high G parallel, A parallel value. For example, your cuprammonium complex will have an A parallel value of 200. So this has blue copper protein has a very low A parallel, which indicates that the coordination geometry around copper in blue copper protein is a tetrahedral one, right? So we can now look at this another electronic absorption spectrum, which simply illustrates this. You can also try to replace copper in metalloprotein, blue copper protein, by other metals and study their function, study their ability to transfer electron, study their spectral property. The red one is your copper one, and these proteins where you replace copper are called reconstituted blue copper proteins. Reconstituted protein, you remove copper, You generate the apoprotein, simple protein, try to incorporate other metal ions. They are all metal reconstituted protein. Among these proteins, it is only cobalt which can take up the tetrahedral geometry. That has got a tendency to take up, behave like copper. As inorganic chemists or MSc students, you should be knowing that cobalt has a higher tendency to form tetrahedral compounds relatively than other metal ions. So people try to replace the copper metal with cobalt, try to study their property. So a lot of activities are there in studying blue copper protein. This is what I told you already. So this is how again you can distinguish between normal aqueous copper EPR, the actual EPR of aqueous copper, how EPR can be measured, and how you can have a blue copper protein EPR spectrum, how you can get the center of these four lines as GPL value, the distance between these two as the parallel value. It's just the EPR spectrum of spinach phosphocyanine, right? Okay. So how these blue copper protein function as electron transfer protein? What is the basic? What is the principle behind this? For example, you can take, I try to illustrate this by taking potassium ferricyanide. What is the potassium ferricyanide? There are 6 cyanide units, ligands bound to ion 3 having an octahedral geometry, right? Now you transfer an electron to the, add an electron to this. What is the splitting, the crystal field configuration? T2G3 Eg2. You can add electron. T2G3 Eg2. You can add an electron and you get ion 2. What is the geometry of ion 2? Again, it is the coordinate geometry is octahedral. So when you add an electron to ion 3 system, ferricyanide to ferrocyanide, the addition takes place, right? You can also remove the electron from ferrocyanide, get ferricyanide. We say the potassium ferrocyanide ferricyanide system, small molecule, is a simple redox molecule used to calibrate your potentiometer. So it's a very reversible one, because when you add an electron, there is no change in the coordination geometry. Simply you add an electron to this T2G3 Eg2 and then get this geometry. So that is why this particular EGOF for the ferrocyanide couple will be positive. Now substitute this one in the delta G equation, you will find that this is a very fragile spontaneous. You can use this Nernst equation to substitute the concentration of ion 3, ion 2 and measure this redox potential. So it all depends on, the redox potential depends on the concentration of ion 3 and ion 2 for simple compound. Now let us go to copper compound. You take an octahedral copper compound, you try to add an electron, right? And see what happens. What is an octahedral copper compound splitting diagram? See, you have an unpaired electron, x square minus y square orbital. What is this x square minus y square orbital? This is oriented towards your x-axis. This is your y-axis. So electron is added here at this orbital here in all copper compound. Already you have a repulsion between, according to crystal field theory, the repulsion between this electron and this electron in the ligand. Right? There is a repulsion. So when you add an electron to this, the already present electron here will try to repel it. So addition of electron is difficult here. Okay? Whereas the XY orbitals are in between the axis. So this is pointing towards the ligand, which is negatively charged. So addition is difficult, right? This is one reason why addition of electron is difficult in normal copper. When you get this octahedral compound is reduced, what is that you see? That is converted into copper 1. You know, copper 1 has a different geometry. Here what dictates that this copper 2 has an octahedral geometry. It is the crystal field stabilization energy that makes this copper 2 stable in an octahedral environment. On the other hand, when you get copper 1, this is not a transition metal now. This is isoelectronic with zinc, D10 system. So what rule decides the coordination geometry around copper 1? So you make use of, for example, the valence shell electron pair repulsion theory. See, here you use crystal field theory, whereas here you use valence shell electron pair repulsion theory. D10 system, so it is a repulsion between these bonds, which dictates the copper 1 to have a tetrahedral. So what has happened here? There are 6 coordinate bonds. 2 bonds are dissociated, decomposed, right? Not only that, 4 bonds are rearranged to get a tetrahedral geometry. Now you can imagine how the addition of electron is difficult, unlike the iron 3 system, because copper compounds undergo structural changes. That is why you have more negative redox potentials with respect to NHE, it is possible, right? So remember, structural changes, coordinative changes, right? They dictate the small molecules to take up, to exhibit, negative redox potential, whereas in blue copper proteins, where you have tetrahedral geometry, right? So that forms the basis of why blue copper proteins are able to, take up electron and give the electron back. So we learned that copper 1 has tetrahedral geometry, copper 2 has octahedral square planar geometry. This is the preferred geometry, copper 2. Preferred geometry for copper 1 is tetrahedral geometry. So fundamental coordination geometry of copper plays the most important role in dictating copper to function as a blue copper. Here I should define some parameters to illustrate the reversible electron transfer in blue copper protein. So you know about the addition of electron. You have a copper rod which is in equilibrium with copper 2 plus ion. You add electrons to this copper 2 will be reduced with electron to copper 1. So what is the yield you get when you add electrons? See, this is the yield. You get current. So current is plotted as a function of the increased potential here. The increased potential is one of the reactants, right here. One of the reactants here, copper is a reactant. So you can simply imagine like this. You get copper 1 as the product. How much copper 1 is generated is measured by the ampere, the current that is we measure, get back. Okay. So here you plot the current as a function of the voltage. Maximum current is obtained at this point, and then the current decreases, and you switch at this one, shoot back to redox potential here. So you add electrons, supply more and more, electrons with more and more energy, reduce it, and then you get back here. So you try to remove this copper 2, or this copper 1. You try to this is minus sign. Okay, sorry. So you try to remove the electron and get back copper. So this can also be reversed. So what is the redox potential in cyclic? This is a cyclic voltammetric measurement, right? You can use a platinum electrode and obtain the cyclic voltammogram for any copper containing system. And here the redox potential can be obtained by taking the average of the EPC, peak potential, cathodic peak potential, this anodic peak potential. Take the average between these two, you get the redox potential. And what is this gap between these two? That is delta EP. That delta EP gives you the facility with which the electron transfer can be done on copper compound. The delta EP is 60 millivolts, right? 60 millivolts. Then we say it is reversible, thermodynamically reversible. According to sequence, you find that this delta EP is 60 millivolts. This 60 millivolts is called one electrochemical decade. In history, we call decade as 60. In 10 years, 60 millivolts is one decade in electrochemistry. So if this is one decade, then the we can get reversible electrochemistry, right? Normally, you do a measurement here for any copper molecule. You switch back this potential at 3 decades, 3 into 60 millivolts, right? These are the techniques we use in measuring the redox potential of copper compounds in our laboratory. So delta E, the E of a normal copper is minus. Okay, when E of is minus, see delta G Pin's equation is positive. So all these simple copper compounds are involved in they involve non-spontaneous reaction. So electron transfer is not reversible. Now you take this copper protein, blue copper protein. Now let us look at, we have learned about the spectroscopy. Now let us try to concentrate on the geometry around copper 2 in blue copper. So this is the process of signing blue copper protein X-ray structure. This structure was solved in 1971, as early as 1971 in Australia, right? But one of the persons involved in solving this structure is an Indian, Professor M.P. Venkatappa. Professor Venkatappa used to visit Madurai Kamaraj University when I was a lecturer there as an examiner for inorganic chemistry. So he was one of the persons involved in solving the X-ray structure of this blue copper protein. He has solved the blue copper protein structure at a pH of 7. He has got the structure of blue copper protein for both copper 2 and copper. So what is that we have found? Copper 2 has a geometry like this. There are 8 ribbons here, organic components, which compel the copper to take up this geometry. So what is the geometry behind this? There are 4 donor atoms. Copper 1 geometry, that also he has figured out that around copper 1 also he has this type of geometry. Now let's focus on this one. See, process signing. What is that you find? Copper is bound by 2 histidine nitrogen, imidazole. Bound to 1 thioether sulfur. I talked about thioether. The lone pairs, there are 2 lone pairs on sulfur like oxygen atom, which is the congener of sulfur in the periodic table. So sulfur is bound to copper. You also have mercaptide cysteinide, which is used here. So what is that you find? More or less, you can just, people who do not know about this, cannot understand this geometry, look at this one. There are 2 imidazole nitrogen from histidine amino acids, right? 2 nitrogen atoms bound to copper. And there is 1 cysteine bound to copper. See, this cysteine is not along the x square, y square orbital, in a different mixed orbital. That is why the copper mercaptan bond is stable. So the X-ray structure is able to explain why this copper mercaptan bond is stable, because this structure is not an octahedral structure or square planar structure. One more novelty is that the copper methionine bond, this is an acidic S minus, so stable, is very much close to copper, 2.1 angstrom units, whereas this bond is at a longer distance. See, 2.85 angstrom unit. So this thioether bond is at a longer distance. See, so now you understand why the A parallel value is very low value. The unpaired electron on copper here interacts with the copper nucleus, that is here itself, very weakly, because it has a tetrahedral geometry. So a tetrahedral geometry determines to what extent the copper nucleus should interact with the unpaired electron. that is the Fermi contact. So the Fermi contact is very weak, right? So here you can very well explain why these blue copper proteins are now able to have very abnormal electronic absorption, absorption. That is mercaptide to copper to charge transfer, which absorbs at 600 nanometer. So it is not the DD band. Copper does. Where does DD absorption takes place? At it's a tetrahedral one. See, any normal copper compound will absorb at 20,000 waves per centimeter. Tetrahedral compounds, you can predict 20,000 waves per centimeter by 2, 10,000 waves per centimeter, or 8,000 waves per centimeter. So our the DD band in blue copper protein should be observed at 6,000 to 8,000 waves per centimeter. So now you see DD band overlaps with the charge transfer band here, mercaptan to copper. So the blue color is because of the charge transfer band from S minus to copper. So now you were able to, with this structure, explain the function, first the structure of this one, and simply also you explain now the function of this. So why are electron transfer occurs very facile way? Simple copper compounds have octahedral geometry, square planar geometry, whereas these tetrahedral geometries have a pre-organized structure, that is a tetrahedral structure stabilized by the organic matrix around this copper, right? So let us focus on this aspirin, another protein that also has a tetrahedral geometry. In fact, it is having a trigonal bipyramidal geometry. There is a very weak bond from the amide bond in glutamic acid, amino acid. So all blue copper proteins have normal geometry, trigonal bipyramidal geometry. Aracinin, that also has tetrahedral geometry. So you can compare prostacyanine geometry and azurin geometry. See, all these bonds are very weak. This is very weak. The amide bond is weak. This bond is also very weak. So now learn that these bonds can be easily fluctuating. This copper sulfur bond at longer distance, copper sulfur bond is also flexible, weak coordination from sulfur, whereas this bond from imidazole is strong. So these bonds can be easily broken up or made, right? So coordination, rearrangement, structural rearrangements can be realized in blue copper proteins at low energy. Stereochemical changes can be imposed on this copper at very low energy. So now you understand these copper geometries, the active site geometries are very abnormal, right? We can compare the geometries like this, in this way, okay? Now see, what is the stereochemical that changes that happen in blue copper protein? The active site geometries, it is halfway between two idealized geometries. What is that? Idealized geometry? Copper prefers to have square planar geometry, right? Copper 1 prefers to have tetrahedral geometry. Copper 2, you have an exception. When you have 4 chloride ions around copper 2, very bulky chloride, that will impose a more or less tetrahedral, not even tetrahedral, a compressed tetrahedral geometry. So remember now that this tetrahedral geometry imposed by the protein matrix around this one is a part of the X-ray structure. So this reactant itself is halfway between square planar geometry and tetrahedral geometry preferred by copper 1. See, it is pre-organized already, right? Copper 1 product, what is the geometry it has? Copper 1, right? Tetrahedral geometry. So this copper 2, copper 1 geometries are more or less the same. There is a small energy difference between these. For any reaction, addition of reaction, you will have a transition state. And you explain this with the help of an entalpic state. Right? What is this entalpic state? This is a pre-organized state, a very, strained state which lies very close to your transition state. So this entalpic state can be there for copper 2 as copper 1 geometries, and they are there just before becoming and entering into transition state. Here you can see for blue copper transition entering the entalpic state, trigonal pyramidal geometry for azurine, tetrahedral geometry. But the entalpic state has still a weaker bond. This is 3.1 angstrom unit or so. Now this is slightly beyond coordinate. So the entalpic state is differing from this state by 4.6 kilocalories per. This 4.6 corresponds to one coordinate bond. This corresponds to your thermal energy also, KT, thermal energy also. So when you supply very small energy to this one, it can transform into enthalpy. This you can understand from the blue copper protein transition state. So when you take square planar normal copper compound, right, your energy activity is very high energy of. So what you get as the product is copper 1, right? Same way if you take up copper 2 in these blue copper proteins, see they are in the pre-organized state already. There's a lot of energy, right? It has a tetrahedral jump. So when it has transition state, this one. So how to reach this transition state? By supply of energy from E prime, energy of activation. See, that is lesser than this one, more or less like your catalyst, right? So the activation energy is reduced. So these proteins are actually present as entalpic state here. So what is this entalpic state? Whenever this curve, the reaction curve, this curve meets this transition state system, curve system, this joint, this is the entalpic state. On this side also we can have an entalpic. So entalpic state has energy less than this transition. So we can easily, already it is a strained situation. It has absorbed a lot of energy, right? So this can go over to transition state and then another entalpic state and then become copper 1 with a tetrahedral joint. So entalpic state theory is used to explain the facile coordinative geometrical changes happening around copper 2. There is a coordinate change, very small, that you can get from the X-ray crystal structure of copper 2 plus cyanine and copper 1 plus 2 cyanine. Whereas for abnormal compounds, you have a different. The energy of activation is high. Here also we can talk about the entalpic state here. See, entalpic state here, which is very high again. For the entalpic state for this compound, synthetic compound, will be very high. This one will be very low for the blue copper. So now you understand how blue copper proteins have a entalpic state different from transition state. Normally, there is a very small energy. You can easily pass through the transition state and gets reduced. So now you know how this geometry is imposed on copper 2 abnormal geometry. That is by the protein matrix. So I understand that the principle behind the function of blue copper protein is imposition of a coordination geometry which is very abnormal. So now let us think about trying to imitate this blue copper protein active site. That's what we have been doing from 1989 or so, 87 or so, from Biomolecular University, our chemistry lab. So what we have done, we have tried to make small molecules, try to get, make the redox potential of copper very low, make synthetic compound, right? So let me try to see. Again, another important thing is, the protein interaction, which makes the electron transfer difficult. See, we have one protein at the copper center is there, another protein in the redox partner. This type of electron transfer between the two copper or ion center is very difficult. You assume that as very difficult because the distance is very large. But that protein-protein interaction, electron transfer between redox partners takes place in a very facile way. So that can be illustrated by considering that one protein like blue copper protein interacts with iron sulfur protein and forms a complex, complex and electron transfer. Though there is a protein matrix here, another protein matrix here, you have more or less like tunneling things which happen here, like this. So we can try to explain all of them by making use of Marcus relations. We have very good textbooks which can explain this reorganization energy. So this is the potential energy diagram for the reactant, for the product, and the delta G is the delta G for these small molecules, whereas proteins have very low delta G. This is a point of intersection. So this is the reorganization energy. But this reorganization energy, which is lambda, in your Marcus relationship, this is here. Okay, reorganization energy is given in terms of this delta G star. Substitute this one here, you will understand that reorganization energy is a controlling thing for electron transfer between proteins. Anyway, this is all very somewhere complex one. These are the two systems where you have entalpic states which are very close to the transition state. So the entalpic state is very low in the case of B here. When the proteins are there, the delta G0 is very low. Whereas for these two systems, the delta G star is equivalent to delta G, very low, and hence the structural reorganization is very low in proteins. These are all for a simple copper compound. You can explain this with the help of this reorganization energy, which is very low for blue copper protein. Okay, now let us think about synthetic complexes of ligands. So we should make sterically hindered ligands which can enforce low coordination geometry around copper. We can isolate copper thiolate compounds, but it is always a challenge. I told you already, copper to thiolate bonds are unstable in small molecules. So how to reproduce the spectral properties in these compounds? So you have to make compounds which have this copper mercaptan bond, or when you don't have copper mercaptan bonded compounds, you can make copper thioether bonds. Copper compound containing copper thioether bonds. Try to discuss the contribution of thioether to copper redox, right? So that type of study will provide insight into the origin of the spectral properties. We try to explain the function as well as the spectral property. And we have tried to generate a copper compound which can transport electron very facile way. So when you generate an oxygen evolving complex, 4 electrons supply to substitute the lithium energy battery, you can now have a blue copper protein model which can take up electron very facile. So anyway, these are all at still miles apart, but then you can try to think like this, try to make a redox partner, synthetic redox partner, to carry this electron from your synthetic leaf to anywhere you like, right? So when we started doing work in our laboratory, so we should think about the structure of this one, geometry, everything. This molecule was made in 1977 or so. We started our work in the US as well as in in biology. In my work in '86 in Drexel University in the US. And I came back to Bhattidasan to do the same type of work, making more compounds, understand this reversibility of this copper compound. This compound, small molecule ligand system, was made in 1977 in the US. That contains two thioether ligands and two nitrogens also. This can be easily made. And now you can add this ligand to copper. So what happens? When you add this one, you isolate both copper 2 compound and copper 1 compound. Same ligand which stabilizes both copper 2 and copper. So such ligand which stabilize both the copper 1 is interesting. When you add thiourea, what did you get? Copper 1. But list the compound added to copper 2, you get a square planar compound. These two nitrogens are bound like this. Two thioethers here are bound like this. So you are able to isolate a copper compound with a very weakly bound perchlorate. So you also get a copper 1 compound which having a tetrahedral geometry. So this is colorless, viscously is colorless, this is colored. So you can separate them very easily with your hand, right? Now, you try to study the EPR spectra, everything, but EPR spectra had a higher A parallel value. So you can do that. From the point of discussion here, I shall concentrate on the E half value. What is E half value? 0.26 with respect to silver plus silver electrode, silver ion electrode. So when you add 530 millivolts to this, or 250 millivolts also you get with respect to normal hydrogen electrode. So E half is positive, higher than aqueous copper. You make tetramonium complex, it's more negative than aqueous copper. So when you make this molecule, it is positive. So substitute this value in the Nernst equation. What is that you find? The redox is fissile. That is why you are able to isolate even copper 1 compound. So copper 2 is easily bleached. Color is bleached. You get copper. Simply you should understand that copper ion is a chemical chameleon. What do you mean chameleon? Changes color depending on the environment. Here is the copper compound, copper metal ion, which can take up a square planar geometry as well as a tetrahedral geometry. So we bioinorganic chemists call this copper a chemical chameleon. When you have a suitable ligand, it can assume any geometry it wants, right? So in synthetic system, we have a very good model which can be used to study the redox. The E half is positive. Not only that, the delta EP at which you conduct this redox at this potential, delta EP is 88 millivolts. That is the difference between E peak cathodic, E peak anodic. So that is 60 millivolts, which you can realize it for blue copper protein. That will be, reversible. Here also you find reversible. When you take aqueous copper, you try to add electron, you will not get back the redox wave, the oxidation wave. You get only the reduction wave. But here you get both reduction wave and the reoxidation. That means you get delta E. So this illustrates that this redox is fissile and it is also reversible. So synthetic compounds can be aimed at to achieve fissile redox and reversible redox, right? So towards that we work. Now in our laboratory, Dr. Usav, my former student, she made a compound similar to this in 1991 or 90 or so. She was our MSc student also. She made a copper ligand system by substituting this by benzimidazole. You cannot replace by imidazole because that is very difficult to synthesize. She replaced this very easily by benzimidazole. Very easy to make compared to imidazole. She substituted these two by benzimidazole and tried to add this ligand to copper 2, and she got an interesting compound like this. So what is that you find? You have a copper compound, right? Two thioethers here, two thioethers, two benzimidazole, So two thioethers bound to copper, two benzimidazole nitrogens bound to. It is not square planar. It is trigonal bipyramidal. So what is that you learn? It is the bulkiness of these two ligands which has imposed on copper an unusual geometry, trigonal bipyramidal. This geometry is almost towards your tetrahedral geometry. So you can use synthetic ligands to make unusual geometries, right? So now you measure your EPR spectra, measure your electronic spectra, compare that with those of the blue copper proteins and all, color, everything. They are not important at this point. You try to measure the redox potential. What is the redox potential? It is 0.563. More positive than the pyridine containing compound is 0.26. But here it is 0.563. More positive. That means, the electron transfer is more facile in this complex. So we were able to achieve a complex, make a complex where the redox is more facile. We go towards that, our aim. More than that, delta EP 0 at 0 scan rate, right? It is 16 milliohm. You get both the reduction wave, copper to copper on wave, as well as copper on to copper 2 wave, and the delta EP is 16 milliohm. You cannot dissolve it in water, you dissolve it in methanol and try to get the cyclic orthomogram. And we were able to successfully publish it in the Royal Society of Chemistry journal, Electron-Transfer. This USA's paper, Electron-Transaction, as early as 1996. So she was an expert in redox as well as in spectroscopy. Then only we can try to make molecules, also yes, synthetic inorganic, and probe this molecule, try to learn about the blue copper protein. Later on, some more students joined me. This was made by another ligand system, see benzimidazole, two thioethers here, another pyridine nitrogen. This ligand system is more interesting. This was made by Dr. Balamurugan, also our MS student here, he was also a PhD student. Now he is at A-Star Laboratories in National Laboratory Science and Technology Laboratory in Singapore. He is making as a good has earned a very big name there as a synthetic chemist, and he made this ligand system. The interesting thing is this ligand system has a square planar geometry. It's a nitrate ion. So two imidazole nitrogen, as in your blue copper protein, another nitrogen also, and another nitrogen. Okay. The beauty is this, when you add this ligand system to copper, see, he got blue color, green colored complex as well as a colorless crystal. See, he had made a ligand which imposed a copper one made a stabilized copper one compound. He isolated, picked up these two complexes separately, and he determined the X-ray structures, and he found that this has a trigonal geometry. This trigonal geometry is very novel. See, copper is known to have only a tetrahedral geometry and a trigonal bipyramidal geometry. We were able to isolate a copper one compound which has a geometry is equivalent to found in fungal lacase, type 4 copper protein. So uncommon geometry for copper 1. Thioether sulfurs here, see, they are not preferred by this ligand, by copper. Altogether different one. So same ligand architecture stabilizes copper 2 as well as copper 1. The beauty of another beauty is here, when you added the ligand separately to copper chloride, that is in the presence of chloride ligand, right? See, he had a tetrahedral geometry, chloride ion bound to copper, right? A tetrahedral copper 1 compound. See, There are two molecules like this having more or less same geometry. So he was able to isolate with this same ligand another complex having copper chloride bond, distorted tetrahedral geometry. The bond angle here is very perfect for tetrahedron. So we have a ligand system which imposes both square planar geometry as well as tetrahedral geometry. It was very much appreciated by peers in the field, and this he was able to publish in Inorganic Chemistry Journal in 2001. So we consider Inorganic Chemistry as the topmost journal, published by inorganic chemists. So we were able to generate a ligand system as in the blue copper protein, which can impose both copper 2 and copper 1 geometry. No wonder he was able to have a very reversible redox and very high copper 2 redox, copper 1 redox potential, right? So people can work toward such systems. And in the literature also, see, he studied all these things, rearrangement with chloride, so you have redox pair. He exemplified very easily with the redox that reorganizational energy and structural changes are very important in determining the fastness with which electron can be transferred. So in the models, you have very high energy needed for reorganization energy, very low energy for minimum, very low reorganization energy that is minimum for blue copper protein. So in synthesis, people have also done a lot of blue copper protein models, right? So this is a very special ligand. Around the same period in Japan, Kitajima, my counterpart, my good friend, he made this type of ligand, hydroxytryptophan, tridentate ligand. I used in my laboratory tetradentate tridentate ligand. On the other hand, he used tridentate ligand. I had the pleasure of visiting him in '93 in Alina Institute, the Tokyo Institute of Technology in Nagasaki. He was making these molecules. So he made a copper 2 compound with a copper sulfur bond, mercaptan bond. He was also unsuccessful in making these copper 2 compounds with tetrahedral geometry. So he made 3 nitrogens, imidazole nitrogen, copper 2, mercaptan bond. So this molecule he isolated, recrystallized this at -25, and the mercaptan bond is 2.16, very close to what we have in blue copper. So when you have employed sterically hindered ligands, you can make sterically hindered copper 2 to impose copper 2 compounds on copper 2 tetrahedral geometry, whereas in our laboratory we used a sterically hindered thioether ligand to stabilize copper 2. And no wonder the A parallel was 70 gas. For our compounds, we got 150 gas as the A parallel, but he was able to have an A parallel of 70. So now you realize 70 gas, can be realized in copper 2 compounds, and recognize that is an index for tetrahedral compound, right? Whenever a synthetic compound is having 70 gas, immediately you can say that it is having a tetrahedral geometry. So this is the knowledge we generated in our laboratory in Bharat University. So what is copper 2 copper redox potential for this compound? Unfortunately, it is minus. It is not plus. But whereas we were able to realize this positive redox potential in our case. In fact, when our students made a lot of thioether benzimidazole compounds to copper 2, sometimes the copper 2 will be bleached. You will not get copper 2 compounds. But immediately colorless copper 1 compounds will be crystallizing. Right? Copper 2 is bleached. That's what I talked about. Copper is a chemical chameleon. It can assume any geometry like this, or trigonal bipolar geometry, tetrahedral geometry, square planar geometry. But people are able to realize this geometry, realize this uncommon A parallel value, but they were not able to reproduce till now. They were not able to reproduce positive redox potentials, but we were able to reproduce positive redox potentials in our laboratory. in Bhartadas. Here is another compound, sterically hindered compound. No? Sterically hindered compound. Beta diketamine. It's a very famous person, isolated this one. Tolman, I met this molecule. He is the editor of Inorganic Chemistry Journal. See, he is now able to introduce thionyl sulfur, mercaptan sulfur. I had the pleasure of visiting him in the US and shared our knowledge with him. So he was able to crystallize this one compound. So you can notice that two imidazole nitrate of nitrogen, one thionyl sulfur, one mercaptan. See, copper mercaptan sulfur 2.176. So it is close to the G parallel value in fungal ligase also. Here parallel is not 70, but lower than that we realized. That is 92 gas. So we have a coordination sphere, copper Ls, S star, and look at the LMCT band. So interesting thing, he was able to get a blue color, and the absorption takes place at 690 nanometer. The epsilon is also high. So he was able to work towards a tetrahedral copper molecule. And redox potential is somewhat higher than normal copper, which is minus 150 millivolts or so. And he was able to get minus 0.200 volts. You just add this 250 millivolts, you get somewhere around more positive redox potential, lower than type 1 copper, right? There's another, so now you try to investigate all these things. What is the knowledge we obtained when we did redox of these molecules? Take, for example, copper imidazole 4 type of molecule. You take aqueous copper, add 4 imidazole, monodentate ligand, you can isolate imidazole. This will have a square planar geometry. Now try to measure the copper to copper 1 redox potential. We found that it was plus 0.4, again positive, whereas aqueous copper is 150 volt. So now we were wondering, so which we used to investigate further. See, we can try to equate this one. The total potential is equal to the aqueous copper plus 4 times the delta imidazole. One can try to see what is the contribution of copper imidazole bond. What is its contribution to redox? So there are 4 imidazole. So we can try to analyze this particular measured value. So we found that this value is additive. This is the basic value for aqueous copper, right? Plus 4 imidazole bond delta E. So we can solve this equation and divide this in 400 millivolts minus 150 divided by 4. You can delta imidazole 50 millivolts. So it is interesting. What is the contribution of imidazole to redox? Aqueous copper, just 50 millivolts. Same way you can make 4 pyridine molecules, copper. You can add 4 pyridine to copper 2 and then find the contribution of pyridine. It will be 60 millivolts, right? For benzimidazole, what is that we found? 120 millivolts, right? So you can make any copper molecule. We have proved that the E half is additive, right? So N heterocyclic will be around 50, N benzimidazole will be 150, 120. Carboxylic acid, suppose you make 4 carboxylic acid molecule here, its contribution is minus 25 million. Thionyl plus 450 million. Thionyl, it will raise the redox potential. So why we have sulfur in blue copper protein? To raise the redox potential, to facilitate the redox. So why do you have N? Why do you have mercaptan? That also raises the lowers the redox potential. Carboxylate lowers the redox potential. Now you can very well explain what is the role of thionyl raise the redox potential. What is the role of carboxylate, phenolate? Lower the potential, but it helps you to anchor copper 2, stabilize the copper 2 in blue copper protein. So remember, these benzimidazoles are better than this one because they can take part in back bonding. You have delocalized pi star orbital, which can delocalize electrons similar to sulfur. So people theorize that it is a sulfur as an empty d pi orbital. The electron we add in blue copper protein to our compounds goes first to the d pi orbital, gets transferred to sulfur and then to copper. So a lot of theories are available. How does it make the electron transfer facile? Right? So that is the theory. That is the explanation we have got from our studies. Right? We can make that redox potential. So we can try to compute the redox potential of any copper compound or blue copper protein. You can compute the redox potential of blue copper protein. So what are the donor atoms present in blue copper protein? We have imidazole, 2 imidazole, thiolate, 1 thioether. Right? Use these values. Additives, delta value, delta EM values. So how can we compute the redox potential of plastocyanine? E0, no? Standard redox potential calculated, the base value, 150 for aqueous copper. How many imidazoles are there? 2 imidazoles. 2 into 0.05 volt, 50 millivolts. In volts is 0.05 volts. Thiolate. What is thiolate? Minus 300 millivolts. Benzimidazole, 150, 120, or something like that. So imidazole 150, thioether 140, right? Thiolate minus 300. So compute all of them, and E0 calculated for plastocyanine is plus 0.09 volts. So what is the best value you use? We have assumed in this calculation that copper has a square planar geometry. That is why the calculated value is lower than the observed value of plus 0.360 for plastocyanine. This value is lower than 360. So now you understand what is the contribution of geometry. So here this is square planar, here this is a tetrahedral, right? Towards tetrahedral. So this redox potential, rise in redox potential is around 250 millivolts. So that is the contribution. Or 0.25 volt is the contribution from the coordination geometry. So one can easily resolve this problem and say the contribution from the redox, the geometry, abnormal geometry is 250 millivolts. So the difference is due to contribution from coordination geometry. So one can try to resolve problems like this by taking up bioinorganic chemistry aspect and then study them. Okay, so we have learned the take-home messages. The electron transfer in blue copper protein in plants is reversible and facile. Now you learn that it is because of the abnormal active site geometry, that is tetrahedral geometry. That is a pre-organized geometry. It is almost in an entalpic state, not in an entalpic state. It is in entalpic state. Now provide an energy of kT, thermal energy, 4.5 kilocalories per mole, equivalent to the one of the coordinate bond. Immediately it goes to the product. Electron transfer is realized, right? So electron transfer in blue copper protein is facile. Bonds are able to transfer the electron obtained from oxygen evolving complex, decomposition of water, takes it to the site of photosynthesis and help in the synthesis of ATP or in the synthesis of organic moieties, NADP get. That is the energy containing organic molecule. So that is also synthesized. Synthesis of ATP is 16 times more useful than getting NADP head. So people are interested in how energy is stored in the form of phosphorus oxygen bond. So I have now exposed you to a lot of fundamentals of bioinorganic chemistry, the role of geometry, role of copper metal bond, and why copper has been utilized in nature, why iron can also be utilized in nature, right? So this is the answer for this question, why blue copper proteins function, why this metallobiomolecule is used as a biological device, metallobiomolecule as a device to fast, facile, and easy electron transfer at a pH of 7 by plants at a normal temperature, room temperature, 25 degrees Celsius. That is how it is planned. So nature has more wonderful things like this as far as the metallobiomolecules are concerned. I shall take up two more molecules, two more cytochromes and this iron sulfur proteins in the next lecture, lecture 2. Thank you for your patient hearing. Thank you. So